Welcome to BioScholar. By the end of this lesson, you'll understand the 15 branches of biology, how it connects to other sciences, and exactly how the scientific method works. So let's start the video. First, what exactly is biology? Science is divided into different branches such as physics, chemistry, mathematics, and biology. Biology is the branch of science that deals with life. It studies living organisms, their structure, their functions, their growth, and their interactions with the environment. Let's understand the word biology. The word biology comes from the Greek language. It is made up of two words. The first word is bios, which means life. The second word is logos, which means to study. So, the literal meaning of biology is the study of life. Life is obviously related to living organisms. And we all know that living organisms include plants, animals, and microorganisms, which in simple language are often called germs. Studying all living organisms under a single subject is not easy. That's why to study life in a better and more organized way, biology is divided into different branches. Biology is mainly divided into three major branches: Botany, Zoology, and Microbiology. Later, these branches are further divided into smaller branches for deeper study, which we will discuss separately. For now, remember this clearly. Botany, Zoology, and Microbiology are the three major branches of biology. First is botany. It is the study of plants, where we learn about their structure, growth, reproduction, and how they interact with their environment. For example, if we take a mustard plant in botany, we study its roots, stem, leaves, flowers, seed formation, and how factors like soil, water, and sunlight affect its growth. Next is zoology. It is the study of animals, focusing on their structure, functions, behavior, and diversity. For example, if we take humans, zoology helps us study body systems, organ functions, movement, behavior, and how the human body adapts to different conditions. The third branch is microbiology. It is the study of microorganisms that are too small to be seen with the naked eye. For example, if we take bacteria, in microbiology, we study their structure, mode of reproduction, living conditions, and their effects on health, disease, and the environment. To understand life more clearly, biology is further divided into different branches, each focusing on a specific aspect of living organisms. The first branch we encounter is morphology. It focuses on the external features of organisms such as shape, color, and patterns, along with how internal organs are reflected in their outer form. In short, morphology helps us understand the overall form and structure of living beings. Next comes anatomy. This branch studies the internal physical organization of organisms, especially humans. It plays a key role in understanding diseases, developing medical tools, and improving healthcare by examining organs like the heart, lungs, and digestive system. Following anatomy is physiology. This branch explains how different body parts function and work together to sustain life. For example, it describes how blood circulates throughout the body, how oxygen is supplied by the respiratory system, and how nutrients are absorbed through digestion. Next, we have histology, the microscopic study of tissues, which are groups of cells that perform similar functions. It helps scientists and doctors understand how organs are structured, how they function, and how diseases affect them. For example, examining heart tissue under a microscope can reveal early signs of heart disease, allowing better diagnosis and treatment. Then comes cytology, the study of cells. Cytologists explore the fundamental structure of cells and their organelles, as well as the mechanisms of cell division. This branch is essential for understanding growth, development, and even cancer, revealing life at its most basic and crucial level. After that, we have molecular biology, which examines biological molecules like proteins, carbohydrates, lipids, and nucleic acids. It studies core life processes such as DNA replication and protein synthesis, contributing to drug development and the creation of genetically modified organisms. This branch provides a deeper understanding of life at the molecular level and drives innovations in medicine and biotechnology. Now let's look at embryology, the study of organism development from a fertilized egg. It investigates how tissues and organs form, identifies birth defects, and supports the development of medical treatments. For example, studying early human or animal development helps in understanding congenital disorders and improving health outcomes. Next, we have genetics, the branch of biology that studies how traits are passed from parents to offspring. Geneticists explore the causes of genetic disorders, improve plant and animal breeding, and investigate heredity and evolution. For example, understanding gene mutations helps in developing treatments for inherited diseases and improving crop varieties. Then comes paleontology, the study of fossils. Paleontologists examine fossilized remains to uncover the evolutionary history of life on Earth. For instance, dinosaur fossils provide evidence of prehistoric life and how species evolved over millions of years, giving us a window into the planet's distant past. Now, let's look at taxonomy, which focuses on classifying organisms into groups based on similarities and differences. Taxonomy helps organize the diversity of life, identify new species, and study evolutionary relationships. Proper classification allows scientists to better understand and explore the complexity of the natural world. Next is ecology, the branch of biology that studies how organisms interact with each other and their environment. Ecologists explore biodiversity, food chains, and the effects of environmental changes. For example, understanding how energy and nutrients flow through an ecosystem helps us protect habitats and maintain balance in nature. Moving on to marine biology, which focuses on life in oceans and other water environments. Marine biologists study ocean biodiversity, discover new species, and work on conservation efforts. For instance, coral reefs support thousands of marine species, showing us how vital healthy oceans are for life on Earth. Next up is pathology, the study of diseases, their causes, and effects. Pathologists look at how microbes, toxins, or cellular changes lead to illness. This branch plays a key role in diagnosing, preventing, and treating diseases. And it helps us understand conditions like infections and cancer more deeply. Next is immunology, the branch of biology that studies the immune system and how it protects the body from diseases. Immunologists explore how immune cells work, develop vaccines, and study autoimmune disorders. This branch helps improve our ability to fight infections and strengthen overall immunity. Then we have pharmacology, which focuses on drugs and their effects on the body. Pharmacologists study how medicines interact with cells and organs, test their safety, and develop new treatments. For example, antibiotics are designed to fight bacterial infections and save countless lives. These are just a few of the many branches of biology. Each branch offers a unique perspective on life, helping us understand the complexity, diversity, and beauty of living organisms. You now know the branches, but a very common exam question is, how does biology relate to other subjects? You can't study the movement of muscles without biophysics, or the chemical reactions in a cell without biochemistry. Let's break down these essential interdisciplinary links, starting with biochemistry. Biochemistry helps us understand what is happening inside living cells at the molecular level. Every activity in our body, from breathing to thinking, depends on chemical reactions. Biology tells us what happens, and chemistry explains how it happens. For example, when we eat food, enzymes break it down into simpler molecules. These enzymes are biological, but their action is completely chemical. Another example is respiration. Inside our cells, glucose reacts with oxygen to release energy, and this entire process follows chemical laws. A third example is DNA. DNA looks like a biological structure, but it is made of chemical molecules arranged in a precise sequence that carries genetic information. So, biochemistry shows us that life is not just about organs and cells. It is also about molecules and reactions working silently inside us every second. Now, let's move to the next field, biophysics. Biophysics helps us understand life by using the laws of physics. Biology tells us what happens in living organisms, but physics explains why and how it happens in terms of force, energy, and motion. For example, think about how your heart pumps blood. The movement of blood depends on pressure and force, which are concepts from physics. Another example is breathing. When we inhale and exhale, air moves in and out of the lungs because of changes in pressure inside the chest. A third example is the movement of muscles. When you walk or run, muscles contract and relax, and this involves mechanical forces and energy transfer. So, biophysics helps us understand living organisms as machines that follow physical laws. Life may look complex, but it still obeys the same rules of physics as the non-living world. Now, let's talk about biotechnology. Biotechnology means using living organisms or their parts to make useful products for humans. Here biology helps us understand living things and technology helps us use them in a practical way. For example, bacteria are used to produce insulin, which is given to diabetic patients. This medicine is made in laboratories using biological knowledge. Another example is the use of yeast in making bread. Yeast is a living organism that helps the dough rise. A third example is vaccines. Vaccines are developed using weakened or inactive microorganisms to protect us from diseases. So biotechnology shows us how biological knowledge is not limited to books or classrooms. It directly improves our daily life, health, and survival. Now let's move to another important field called bioinformatics. Bioinformatics connects biology with computers. Living organisms, especially humans, have a huge amount of information stored in their DNA. Studying this information manually is almost impossible, so scientists use computers to help them. For example, computers are used to study DNA sequences and identify genes related to diseases. Another example is comparing the DNA of different organisms to understand evolution and relationships between species. A third example is using computer software to predict how proteins are formed and how they might behave inside the body. So bioinformatics helps scientists handle large biological data quickly and accurately. It makes modern biological research faster, smarter, and more efficient. Let's talk about biostatistics. Biostatistics is the use of mathematics in biology. In biology, scientists collect a lot of data, and biostatistics helps them understand what the data actually means. For example, when doctors study how a disease spreads in a population, they use numbers and graphs to analyze the data. Another example is testing medicines. Scientists use biostatistics to check whether a new drug is effective or not. A third example is population studies where biostatistics helps in understanding birth rates, death rates, and the growth of populations. So, biostatistics helps biology become more accurate and reliable. It allows scientists to draw correct conclusions instead of guessing. Now let's discuss computational biology. Computational biology uses computers to understand and predict biological processes. Sometimes real-life experiments take too much time or are too complex. So scientists use computer models to study life processes. For example, scientists use computers to predict how a disease might spread in a population. Another example is studying how proteins fold into specific shapes inside cells using computer simulations. A third example is modeling how cells grow or how organs develop over time. So, computational biology helps scientists test ideas safely and quickly before doing real experiments. It saves time, reduces cost, and improves accuracy in biological research. Now, let's talk about bioeconomics. Bioeconomics connects biology with economics. It helps us understand how biological resources are used wisely so they are not wasted or destroyed. For example, managing fisheries requires knowledge of fish populations so they are not overfished. Another example is agriculture, where biological knowledge helps farmers increase crop yield while keeping costs low. A third example is protecting forests and wildlife, where economic planning is needed to balance human needs with environmental conservation. So bioeconomics teaches us that living resources are valuable and limited. By combining biology with economics, we can use nature responsibly and ensure sustainability for future generations. Now let's discuss an example of interdisciplinary collaboration in science. Imagine you're born with a book inside you. Not a storybook, not a diary, a manual. Every page of this book decides something. How tall you'll be, what your eyes will look like, how strong your heart is, even what diseases you might get. For most of human history, we knew this book existed, but we couldn't read it. Doctors could see the effects of disease, but not the instructions behind them. Scientists knew DNA carried information, but they didn't know what that information actually said. So in 1990, something bold happened. A group of countries came together and said, let's stop guessing. Let's read the whole book. That mission became the Human Genome Project. The goal was simple in words, but huge in reality. To read every single letter written in human DNA, all 3 billion of them. Think about that. 3 billion letters A, T, G, and C spread across 23 pairs of chromosomes inside every cell of your body. Those letters are not random. They are instructions. Instructions for building muscles, for growing nerves, for repairing skin, for fighting infection. Scientists had to cut DNA into millions of small pieces, read them using machines, and then use powerful computers to put the whole thing back together, like rebuilding a shredded book page by page. It took years. It took thousands of scientists. It took countries working together. But they weren't doing it out of curiosity alone. They did it because behind every genetic disease, every inherited cancer, every unexplained disorder, there was a broken sentence somewhere in that book. And if you can find the broken sentence, you can start fixing the problem. That's what the Human Genome Project really was. Not just a science experiment, but humanity finally learning how to read itself. After deciding to read this book of life, the next question was obvious. And why would anyone even try? This wasn't a small lab project. Reading 3 billion DNA letters was going to take machines, money, and years of work. Many people at the time said it was impossible. Two main groups stepped forward. One was the U.S. National Institutes of Health, the part of the government that deals with human health. The other was the U.S. Department of Energy, which sounds strange until you know the reason. After World War II and nuclear testing, scientists were worried that radiation might damage human DNA. So, the Department of Energy wanted to understand what normal human DNA looks like so they could detect what radiation changes. So, these two groups joined forces. Soon other countries came in. Britain, Japan, Germany, France, China. It became a global mission. But there was a twist. At the same time, a private company said, we can read the human genome faster and we will own it. That meant human genes could become private property, something to be bought and sold. The Public Human Genome Project made a firm decision. The human genome belongs to everyone. So, this was no longer just a science project. It became a race not just to finish first, but to decide whether the basic code of human life would be free or owned. And that's what pushed the project forward with full force. Now the mission was set. The book existed. The team was ready. So the next question was, what exactly were they trying to read? Inside every one of your cells are 23 pairs of chromosomes. These chromosomes are made of long strands of DNA. And DNA is written in just 4 letters, A, T, G, and C. Those 4 letters form words called genes. And those genes tell your body how to make proteins, the things that build and run everything in you. The Human Genome Project had 3 big tasks. First, find every gene in the human body. Second, read the exact order of those DNA letters. Third, map where each gene sits on the chromosomes. But this was not like reading a normal book. DNA had to be cut into millions of tiny pieces, copied again and again, read by machines, then reassembled by computers. Imagine tearing a giant book into shreds, scanning every piece, and then asking a computer to put it back in the right order. That's what scientists were doing on a scale never tried before. And this is why the project had clear goals. Not just to read DNA, but to store it, analyze it, protect it, and make sure the knowledge could be used by doctors and researchers everywhere. This was the moment biology stopped being small scale and became big science. Now, something unexpected started to happen. As the pages of the DNA book were being put together, scientists began to notice patterns, and some of them were shocking. For a long time, people believed that humans must have an enormous number of genes. After all, we are complex. We think, feel, create, and suffer from thousands of diseases. But when the Human Genome Project counted them, the number came out to only about 20,000. That's not much more than a mouse. It meant something important. Being human is not about having more genes. It's about how those genes work together. They also discovered that all humans are almost identical at the genetic level. More than 99.9% of your DNA is the same as every other human on Earth. So skin color, height, eye shape, even disease risk, all of that comes from a tiny fraction of our DNA. And then came the medical breakthrough. Scientists could now look at the genome and say, this gene is linked to breast cancer. This one affects heart disease. This mutation causes a child to be born with a genetic disorder. For the first time, disease had a map. Doctors were no longer just treating symptoms. They could see where the problem began, inside the code itself. That is when medicine quietly entered a new era. Once the human genome was read, medicine was never the same again. Before this project, doctors treated people based on averages. If a drug worked for most people, it was given to everyone. If it failed, no one knew why. But after the Human Genome Project, doctors could look at a person's DNA and see differences that mattered. They could predict who is more likely to get cancer, who might develop diabetes, which medicine will work, which medicine might cause harm. This is called personalized medicine, treatment based on your own genetic code, not guesswork. The project also made genetic testing possible. A simple blood sample could now tell parents if their child carried a genetic disorder. Cancer could be detected early, even before symptoms appeared. And then came something even bigger. If you can read DNA, you can also edit it. That idea led to tools like CRISPR, where scientists can fix broken genes, like correcting a spelling mistake in a sentence. The Human Genome Project didn't just explain disease. It gave humanity the tools to fight it at the root. Medicine stopped reacting and started predicting. The Human Genome Project didn't just read our DNA. It changed how we understand life itself. For the first time, humanity looked into its own instruction book and learned how to fix its mistakes. And this was only the beginning. The book is open now, and the future of medicine is being written from its pages. Why does one plant grow faster than another even when both receive the same sunlight and water? Or why do some people get sick from the same food while others remain perfectly fine? Questions like these may seem simple, but they are the starting point of every scientific discovery. When scientists face such mysteries, they don't guess, they investigate. The organized process they use to find reliable answers is called the scientific method, and it is the foundation of biology. Through this method, curiosity turns into evidence and questions turn into knowledge. The following are the steps of the scientific method. Every scientific journey starts with noticing something unusual. When a scientist observes a change in nature, curiosity is triggered. This curiosity is then turned into a clear question that can be investigated. This step is called recognition of a problem, the moment when an observation becomes the starting point of scientific inquiry. For example, a biologist might notice that plants in a particular area are growing taller than usual. Instead of ignoring this, the biologist asks, what factors are responsible for this increased growth? That single question becomes the problem to be studied, and it marks the first step of the scientific method. After recognizing a problem, scientists move to the step of observation. In this stage, they carefully collect information about the problem using their 5 senses, tools, and previous scientific studies. Observation helps scientists understand what is really happening before making any explanations. Observations can be of 2 types: qualitative and quantitative. Qualitative observations describe qualities that cannot be measured with numbers, such as the color of a flower or the texture of a leaf. Quantitative observations involve measurements and numerical data, such as counting the number of birds in a tree or measuring the height of a plant. Because quantitative observations are measurable, consistent, and recordable in numbers, they are considered more accurate and reliable for scientific investigations. Based on the observations, scientists then develop a possible answer to the problem. This proposed explanation is called a hypothesis. It is a tentative answer, meaning it may or may not be correct, but it is always based on evidence, not guesswork. To build a strong hypothesis, scientists also study books, research papers, and previous experiments to understand what is already known. A scientist may form more than one hypothesis for the same problem. However, every good hypothesis has some important features. It must clearly propose an answer, match the observations, and most importantly, it must be testable through experiments. A scientific hypothesis must also be open to being proven wrong, because science advances by testing ideas, not by protecting them. Once a hypothesis is formed, scientists predict what results should appear if the hypothesis is correct. These logical predictions are called deductions. Deduction helps scientists plan experiments by clearly stating what they expect to observe. Deductions are usually written as if and then statements. For example, if the plants are growing taller because the soil contains more nutrients, then adding the same nutrients to other soil should also increase plant growth. Similarly, if better sunlight is responsible for faster growth, then plants given equal sunlight should grow at the same rate. These deductions guide scientists toward the next step: experimentation. Experimentation is the most important step of the scientific method. In this step, scientists test their hypotheses to find out which one is correct. A well-designed experiment helps accept the correct hypothesis and reject the incorrect ones. Scientists often repeat experiments and form new deductions to confirm their results. To perform a fair test, scientists set up 2 groups, the experimental group and the control group. The experimental group is the one in which the factor being tested is changed. The control group is the group kept at a normal level for comparison. This comparison helps scientists clearly see the effect of the tested factor. For example, to test whether carbon dioxide is necessary for photosynthesis, 2 similar plants are used. One plant is not given carbon dioxide while the other is given carbon dioxide. If photosynthesis occurs only in the control group, it proves that carbon dioxide is necessary for the process. After performing the experiment, scientists carefully record the results. This step is called data collection. Data may be collected in the form of measurements, numbers, observations, tables, or graphs. Accurate data collection is essential because scientific conclusions depend entirely on reliable data. Data can be quantitative, such as measuring plant height or counting leaves, or qualitative, such as noting changes in color or appearance. Scientists record all data honestly without altering or ignoring results, even if they do not support the hypothesis. After collecting data, scientists carefully examine and interpret it. This step is called analysis. During analysis, the collected data is compared between the experimental group and the control group. Scientists look for patterns, differences, and relationships to determine whether the results support the hypothesis. Tables, graphs, and charts are often used during analysis to make the results easier to understand and more reliable. Based on the analysis, scientists then draw a conclusion. In this step, they decide whether the hypothesis is accepted or rejected. If the results support the hypothesis, it is accepted. If not, it is rejected or modified. A conclusion is always based on evidence, not personal opinion. Sometimes conclusions also lead to new questions, starting the scientific method all over again because science never truly ends. After reaching a conclusion, scientists share their findings with other scientists. This step is called communication with the scientific community. Results are communicated through research papers, journals, conferences, seminars, and presentations. Sharing results allows other scientists to review the work, repeat the experiment, and verify the findings. This process helps remove errors, improve accuracy, and expand scientific knowledge. Without communication, scientific discoveries would remain personal opinions rather than accepted scientific facts. When the same hypothesis is tested repeatedly by many scientists and consistently supported by strong evidence, it may develop into a scientific theory. A theory is not a guess. It is a well-tested explanation of natural phenomena based on observations, experiments, and verified data. Scientific theories explain how and why things happen in nature. They can be modified or improved if new evidence is discovered, but they are never accepted without strong experimental support. Examples include the theory of evolution and the cell theory. A theory represents one of the highest levels of scientific understanding. When scientific explanations become well established, science also identifies consistent patterns in nature that can be described in the form of laws. A scientific law describes what happens in nature under specific conditions. It does not explain why it happens. Scientific laws are based on repeated observations and experiments and are often written as statements or mathematical relationships. They are reliable but limited, applying only where the same conditions exist. For example, Mendel's laws of inheritance describe how traits are passed from parents to offspring. From a simple question to a tested law, science follows a disciplined path. Each step of the scientific method builds on the one before it, ensuring that knowledge is based on evidence, not assumptions. This systematic approach has allowed biology to grow from simple observations into a powerful science that explains life itself. If you found this video helpful, don't forget to like it, share it with your friends, and subscribe to the channel for more simple and clear explanations of biology. Keep learning, stay curious, and I'll see you in the next video.